And so this is, it really kind of falls on the heels of what Sam just mentioned, and he did kind of go over some of the difficulties. I will throw in this slide because this slide was made by someone who left our lab about a year ago, but he said this picture reminded him of what I would look like if I had hair. And so I am quite comfortable holding things that usually shock me. The electrospray source on the mass spectrometer runs at somewhere between 22 and 2500 volts, and I shock myself with it once a week. But it's mostly a static shock, so it's not too bad. This looks maybe a little more dangerous. Um, and so, what I'm going to kind of go through here are just common things that we see that go wrong, and maybe uh, how we can help it. And so, experimental design is, Sam touched on it, but it really is probably the biggest source of problems. Uh, I'll go over a few rules about sample processing and then troubleshooting for sample prep. Um, it really, the sooner that we can get involved, the better off we're going to be. And so if if we can uh, communicate with the researcher before the experiment's done, it's so much easier because adding one more replicate, even though it's a lot of work, um, is much more work after the experiment's done, right? And so there was a good quote that our bioinformatician used to use, and I'm going to mess it up, but essentially they would ask them to diagnose a problem, much like you'd uh, ask a mortician to diagnose a problem of a person, and then in hopes of trying to revive them, right? So that's a little late. You'd rather have the surgeon diagnose that problem, or better yet, your primary care physician a year before. So um, uh, communication, it's like location in real estate. So um, if we know more of what you're doing, it helps us. And so sometimes these consults with Zoom now, we spend more time I think on Zoom than we do in the lab on some days, and sometimes we feel like we're getting too much nitty gritty detail about the experiments, but for the most part, we don't get enough. The more we understand of what you're trying to do, the more we can help you. And we're definitely not experts in biology. I myself am the self-proclaimed village idiot of biology, never having had a college biology course, but I've learned a few things along the way, and so um, it definitely helps if you're on the same page. And then involving the either a biostatistician from your institute, or the bioinformatic consultants, here within our core, getting them involved early is paramount. Sam talked about the biological replicates, and not too long ago, if you did a duplicate, that was great, right? You could do two, that was awesome. Now you have to do three of anything, and really you can only get away with having three if it's a sample that is highly reproducible. A cell culture, triplicate is probably fine for a pilot study. With animals, and Mike Kinter will probably chime in on this. 5 is the bare minimum for an animal study, and Mike would prefer you have 10 or 15, just because of the variability in animal to animal. And so also knowing if the sequence database is available, it changes kind of what experiments can be done. We can do things that are that are not sequenced. Homology is good enough in proteomics for identifications. Much harder for a characterization study. But the biggest problem is statistical power, and so statisticians can help you power a study, but those are really based on your perception, right? So you expect to see a twofold change, and so you need an N of 3 to see a 2-fold change. But what you see is a 1.5-fold change. And so now it may not be powered enough. So, you know, sometimes you're wanting to do a preliminary experiment and you've picked a dose that's simply too small. It doesn't give you enough of an effect. And so, you know, it's always good to have some type of readback or phenotype or something on your experiment, you know, before you go through the expense of doing a proteomic experiment to realize, wow, I've got 2 really good sets of controls here, you know, because my dose wasn't high enough to actually induce a change. And then another issue, and our bioinformaticians pull their hair out with this one, is when you have too many variables and you're trying to compare too many different things. And it's, you know, it's best to be able to answer one question well than 5 questions sort of well. So for sample processing, you know, these are pretty straightforward. You know, starting with a higher amount of protein always helps. You know, Sam kind of got into this. You know, we don't need 100 micrograms to start with, but if we have that amount, we know that it's likely not going to fail. And so one thing that this runs into is maybe you've got 10 samples, and we've got 100 micrograms for 9 of them, and for 1 of them we have 20 micrograms. Well, now we can do 2 things. We can throw out 1 sample, which if you've done triplicate analysis, well, now there's a problem, right? So now we've thrown out 1 of your 3. So having an N of 5, you know, helps that problem. But if we can't throw it out, well, now the only thing we can do is only use 20 micrograms from all the other samples, and so that's going to decrease the depth that we can see. The mass spectrometer is going to identify the abundant proteins first, and then the abundant protein second, and then the abundant protein third, and it's going to take a while before it gets down to lower abundance proteins. And so the more protein we have, the better off we're going to be in detecting those rare proteins, and everybody's always interested in the lower abundance proteins. Time delay in processing samples, and even difference in you know, the person in the lab that acquires them. You know, because you could definitely see an effect if, you know, if one person in your lab was you know, dissected your mouse livers one week, and someone else you know, dissected livers the next week. You know, having consistency in processing the samples will really reduce the batch effects and allow you to have a tighter in-group variability, which allows you to see smaller changes between groups. On protein quant, there are lots of different ways to get to a protein quant. We're kind of continually amazed that we get protein quants that are just, you know, really nowhere near reality. And oftentimes this comes because there's, you know, something in the buffer that's interfering, and you're measuring that. You know, we get protein quants from someone's done a lysate, they send in protein quants, and they're all completely identical. You know, either they're really, really good, or they've measured, you know, some component of their buffer, and it's usually that. And so, you know, making sure you do a buffer blank, and these are, you know, kind of common sense things, but it's amazing how often that it really kind of comes up. Again, something incompatible in the lysis buffer, you kind of think we're being overboard asking for the full list of buffers in your pull downs, and we've had people that I guess they think you know, the secret sauce in their pull down is a trade secret, and don't want to tell us everything. You know, we're not going to run to your you know, your scientific competitor and show them the components of your buffer. But what we want to see is to make sure that there's not a 10% tween solution in that final rinse that, you know, maybe makes your data not stick to the tube when you dry it down, but for us is really going to kill us when we have low level sample. Having amine containing buffers if we're going to do a TMT experiment, because the TMT reagents react with primary amines, and so things that we just need to know. Shipping with dry ice, especially with the issues we've had with FedEx around the holidays, and then now with weather, making sure that there's enough dry ice in the box. Having a big box with 1 pound of dry ice is not going to get you very far. So, pack it full. Information on the protease inhibitors and phosphatase inhibitors as well. So I mentioned the detergent, or with SDS, having too much is actually an issue. If we're doing the fast protocol, we're using the filter, we have to dilute this up, and we just can't, starting with 4% SDS, we can't dilute it below the micellar concentration of SDS. So, we end up getting, multiple milliliters of solution. And so it's just not, not feasible. We have people that sometimes want to run gels, and this is getting less and less common. The mass spectrometry is really taking hold, and we're able to do things in these data independent runs that previously we had to do with gels. But we still see this. But for gosh sakes, don't pour your gels. I know you can pour them cheaper than you can buy them, but the differences in contamination and cross-linked cysteines and modified amino acids from acrylamide, it makes all the difference in the world. Save up, and if we have to give you a price break so you can buy a gel, we'll do that. Something. Please don't pour your own gels. Kamasi staining instead of silver staining a gel. We don't need the stain. Sometimes you may want the stain just so you can see, see your band. If you want an image, run a different sample and silver stain that for your records. We just want enough Kamasi to be able to see the edge of the lane. Typically, the mass spectrometer is going to be a lot more sensitive than any stain. Use clean containers. Don't stain it in a container that has been used to stain gels in your lab for 20 years and is sitting out with dust on the shelf. Most of dust is human keratin, and so the human keratin will be a definite distraction for the mass spectrometer to identify low abundance proteins. Wearing gloves. Putting on new gloves to cut, cutting out with a new razor blade or scalpel. Take a picture. You can put these on a scanner, or pictures with iPhones work out reasonably well now too. If you want to cut the gel, putting it in a PBS at 4 degrees is fine. Sometimes if you freeze these, the gel will actually disintegrate into smaller pieces, and it's an issue with these little gel shards for clogging the instrument later. And so sometimes if you have proteins can't be solubilized in SDS, even without heat, urea, thiourea definitely can help. And there are other methods that we can talk about. But before you use some odd buffer, again, contact us. For fat-containing samples, we need to know this, and we'll do typically the chloroform methanol extraction does a pretty good job of removing fats and lipids. Or we can do an acetone precipitation. Sometimes with an acetone precipitation, it's an issue of getting that pellet resolubilized. And so this is where we follow my famous rich rule of half. The mass spectrometer is really sensitive at the end. So in the end, whatever we're injecting, we're typically injecting 1 microgram of peptides at a time. And so if you back it out, we only need a few micrograms of material to start with. Well, why in the world do we say we want 100 micrograms? Well, it's so we can have room for failure. And so if we have enough protein in the lysis, then we can do a digest. If it fails, we typically still have enough material that we can repeat that, because the last thing you want is to have an underpowered study with an N of 3, and one of the samples gets lost because the filter fails on a filter-aided digest protocol, or someone in the lab makes a pipetting error, which still does happen, not very often, but when there's enough material that we can overcome this, it's a whole lot nicer for everyone involved. Finally, toward the end, so we're doing looking at data collection and analysis, QCing the data, looking at principal component analysis is a great way to look at kind of how the data sits, and do your replicates sit together? Is there something that a confounding feature about your data that maybe you didn't know before? Something confounding you're seeing, a male versus female effect, or a day in cell culture, or a person in the lab who was in charge of growing the cells over that weekend difference, or something like that. And looking at a Venn diagram for the missing values in the data. Missing values are something that aren't as much of an issue when we're looking at data independent analysis, because that type of experiment gets you a value for everything. But in larger TMT experiments, when we have more than 10 samples, that can be an issue. And so make sure you're not just seeing an effect because one protein is detected in one experiment and not detected in another. So. All right, I ran through that pretty quick, but a lot of it is, take-home messages. Don't use tween.